I'm very excited to introduce today's speaker. We have Professor Anne McArdle, Professor of Musculoskeletal and Aging Science at the University of Liverpool. Professor McArdle is a leading researcher in skeletal muscle aging with a particular focus on understanding why muscle mass and function decline as we age and how factors such as inflammation, mitochondrial dysfunction, and cellular stress responses contribute to this process. Her work spans both fundamental and translational research, including the MicroAge program, which uses experiments conducted aboard the International Space Station to study microgravity as a model for accelerated muscle aging and identify strategies to preserve muscle health. Professor McArdle has held prominent leadership and editorial roles within aging and physiology research communities and has presented her work internationally. And we are very, very excited to have her here. Please join me in welcoming Professor McArdle and take it away whenever you're ready.
Thank you. Thanks for such a generous introduction. And thank you for the invitation to present our work to you here today, which is going to focus, as you say, on our MicroAge studies, which are looking at muscle loss in microgravity and what we can learn from that in terms of the development of sarcopenia here on Earth. So I'm based at the University of Liverpool in the UK, and for some time now, apologies, I'm not able to advance the slide. No, I'm not sure what's happened here. Oh, there we go. Apologies for that. So for some time, we've been interested in the mechanisms by which muscle degradation occurs in a whole host of situations, but more recently, particularly focusing on aging. And so what I'm going to talk to you today about is why we're interested in aging, why it's a problem, a little bit about what we know about muscle loss with aging at the moment, and then what we can learn from some of the studies in space. So why aging? Well, if you look at this slide here, you can see that back only in the 1800s, life expectancy, you can just about see in the bars at the bottom there, the average global life expectancy was 29 years. 1950, if we move up to 1950, you can see that's gone up to the 40s, and some more significant improvements in developed countries up to 60s and 70s. And then life expectancy in 2015 here considerably increased globally to 71 and up into the late 80s for some of the some countries. And that's a major success story for global aging. It's mostly due to medical advances and antibiotics, vaccines, and so on. But it's quite a striking effect over a relatively short period of time. And I think one of the interesting calculations that's been undertaken by Professor Tom Kirkwood at Newcastle here in the UK is that a child born today will have, on average, 5 hours longer lifespan than a child born yesterday. And that's the rate at which aging lifespan is increasing. The problem that and so what we're seeing is that the population age 65 and over in the UK and in other countries is expected to increase by almost a third in the next 20 years. Problem that we have is that brings with it some disabilities. And so if you look at some data here from the Office for National Statistics, the number of years that people can expect to spend without a disabling health condition is actually not even staying steady, it's falling. And so if you focus on women here, back only as far back as 2006, 2008, you could expect to have disability-free life expectancy to about the age of 65. Move on for less than 20 years, and that's decreased to 61. So we're seeing failing health in large numbers in our older population as that population increases as well. So we're seeing more need for residential care, hospital care, and so on. And what we'd really like to see, we're not particularly interested in extending lifespan substantially, but what we want to see is improved health span. We need our older people to be able to undertake what they want to do, exercise, play with grandchildren, and so on. Now, there's an additional complication here, is that because of that need for care, every day around 600 people in the UK are having to give up their work to care for others. So there's a double hit here in that we have healthy people also having to give up their work. So why are we interested in muscle? Well, one of the most common problems leading to increased frailty and immobility is loss of muscle mass and strength. And you can see that on these images on the right-hand side. So somebody in their mid-40s in the top image has enough muscle to undertake everyday tasks to do what they need to do to move around. This lady in her mid-80s has lost about 50% of her muscle mass, and she's right on the threshold of being able to look after herself, struggling to walk upstairs to pick up a pan of water and so on. So what's happening to that muscle? Well, we're seeing a loss of muscle bulk, as you can see here, and a loss of individual muscle cells called muscle fibers that run the length of the muscle pretty much. And so if you look on the graph on the left-hand side here, muscle cross-sectional area, you can see that from the age of about 40 to 50, we see a decline in that muscle area. So this lady here, down to about half of the muscle mass she had when she was in her 30s and 40s. The main reason for that is that you lose completely muscle fibers, muscle cells. And so you've total number of muscle fibers shown from some very neat work undertaken by Jan Laxell's lab, again, has dropped significantly. The additional problem that of these muscle fibers is that the ones that remain are also smaller and they're weaker. Okay, so you might say, well, can't we just tell all older people to exercise to prevent muscle loss? Exercise at any age is undoubtedly good for us, particularly for the cardiovascular system. But the only sort of exercise that's really beneficial to older people is resistance exercise, shown here. And there are lots of reasons why our older population can't undertake that sort of exercise, with joint issues, with cognitive issues, mobility issues, and so on. The other problem is that exercise isn't the full answer. So even if somebody exercises for their whole life with resistance exercise, we still see loss of muscle mass. So these data show what happens. You've seen this before. What happens to our muscle strength from the age of about 40 down to here, the age of nearly 90, from a healthy sedentary population. The line on the top shows that even if you're a veteran weightlifter, so you've undertaken weightlifting for the whole of your life, you decline at the same rate, your muscle strength. The difference is that these veteran weightlifters are using what muscle they have left optimally. And it would be amazing if we could take these healthy sedentary people and optimize their muscle strength in the same way. If we do that, you'd take them back in terms of strength to their 40s or 50s. So that would be a really interesting approach to take. The problem we have is that muscle of older people is resistant to any sort of exercise or any sort of intervention. We call this anabolic resistance. And so you can only really improve a little bit this muscle strength in older people with resistance exercise. That doesn't mean you shouldn't exercise, because that in itself would take you back 10 years if you can exercise as it stands in terms of our understanding of muscle loss in old age. It would be great if we could understand why these muscles are resistant, and we still don't really understand this. And that's where a lot of our work has gone to in the past. And I'll just give you a very brief overview of where we are with this work before we move on to the exciting space work. So we know that if we exercise muscles, we get an acute generation of reactive oxygen species or free radicals during the contractions, and that activates adaptive responses to the muscle that causes this hypertrophy, this building of muscle that we see in younger adults. These data are in mice, and on the right-hand side are a human equivalent study. So if we undergo an acute isometric contraction protocol, we see particularly interested here in this rise in heat shock proteins or chaperones, which are really involved in the chaperoning of newly synthesized proteins that build muscle following contractions. So here we've got HSP60 and HSP70. They're controlled by heat shock factor 1, and some of the other responses that we see are controlled by other transcription factors such as AP1 and NF-kappa B. So to get this acute response in young muscles, we see an acute activation shown in these EMSAs of heat shock factor, transcription factor 1, AP1 and NF-kappa B. And it's important to remember that that's an acute response that falls off very quickly. And then we get this protein and anabolic response. I haven't got time today to talk about the detailed mechanisms for activation of that response. And this is where we think that the problem is in terms of our older muscle. So we think that the activation process is through ROS production by an NADPH oxidoreductase at the plasma membrane, which starts a cascade of redox relays that activates these transcription factors that you can see activated here. So what goes wrong in old muscles? Well, some of our data here is shown using isolated viable fibers that we can take from adult and old mice. And in this situation, we can look exclusively at the muscle fiber, a viable muscle fiber, but it's from an old mouse. So it has a lot of the characteristics of an old muscle in culture. So it's a really neat way of studying aging in muscles. We can load these muscles with different dyes that show generation of reactive oxygen species. So if you focus on the open bars on this graph, you can see that if you take muscle fibers from young mice, they've got relatively low ROS levels. If you then exercise them, you see this acute increase, and that's the signal for these activation of transcription factors and anabolic response that we see in young muscle. If we look in the muscle fibers from old mice, you can see that ROS generation is already elevated, and you can't then see a further increase with contraction. So it's a blunted response at the level of that ROS generation. And in contrast to the activation in young muscles, which we think is through this plasma membrane enzyme, this chronic increase in ROS in fibers of old is likely to be due to mitochondria, so produced by mitochondria. And that work's being undertaken by a whole host of studies in our lab and in others using different inhibitors. So then if we look at old muscle and the production of heat shock proteins and transcription factor activation, you can see that you saw these data before, the HSP25 content of adult muscles in the gray bars on the left-hand side, and you see an acute production of heat shock protein, 25 in this instance, but lots of other HSPs. You don't see that increase in old. So we really have got this blunted response. If you look at the EMSA, if we focus on NF-kappa B here, you can see that if we take the adult quiescent muscle and contract it, you saw those data earlier. We see increased activation of the transcription factor NF-kappa B. What we see in old is a chronic activation of NF-kappa B to start with, probably because of that chronic ROS production by mitochondria, and then we can't see an increase in transcription factor activation after that. So that chronic increase in quiescent muscle is very different to the acute activation of NF-kappa B that's needed for anabolic responses. I'm just going to tell you about some data on HSP10 here, which links into our space studies. So we then started to put back some of these heat shock proteins into muscles. So we overexpressed them throughout life in animals and looked at the effect of this. So HSP10 is a mitochondrial heat shock protein or chaperone. We saw that we got increased expression in the mitochondrial fraction of our muscles. What we saw was preservation in terms of protein oxidation in the mitochondria. And these work are all published, so I'm just going to talk very much in brief. But the take-home message really was that the normal fall in muscle function that we saw in wild-type animals was not evident when we protected the mitochondria of the mice. And we could mimic that with an inducer of HSPs, 17-AAG as well. So we know that aging now attenuates this adaptation in contractile activity. I'm just going to focus back on this chronic activation of NF-kappa B. If you do the calculations, and we all think that the systemic inflammation that we see in old age comes from immune cells, and it undoubtedly does. But actually, we've now shown that a lot of the pro-inflammatory cytokines, shown in this next slide, that you see in the serum of animals and humans comes from muscle. And so this is really bad. We don't want muscle to be producing these cytokines. So we don't want that chronic activation of NF-kappa B. What we know is that if you're inactive, your muscles do produce some of these proteins as well. And we wondered how many of the problems that we see in old age are due to this inactivity. So is inactivity causing this chronic activation of NF-kappa B, which is partly responsible for the blunting of the adaptive responses? So we know that inactivity causes some of the chronic inflammation in older people that we see. Older adults spend 60 to 70% of their day in sedentary behavior, and that has really negative outcomes. So we really wanted to separate aging from this, per se, from this sedentary behavior. And that's where the space studies came in. It's really hard to study sedentary behavior in old people. We don't want to make them more sedentary. And also undertaking invasive studies is particularly difficult. So space studies. We were very keen to know when we noticed what was happening to astronauts when they came back from the International Space Station from several months in microgravity. There's clearly an issue in microgravity with muscle loss because there are lots of ways that the astronauts are trying to overcome this. And current best practice is some of the exercise protocols that you can see on this next slide. But despite this, you can see that, and this is some of the recent astronauts, Tim Peake in the middle from the UK, they've still lost about 30% of their muscle mass after 6 months in microgravity. The other thing is, despite this happens despite the exercise, so they've also got this blunted response to exercise, so the anabolic resistance that we see in older people. Thankfully, when the astronauts come back from the space station, because they're young, they can rebuild their muscles. But we wondered whether there was a common pathway to what's happening in aging and what's happening more rapidly in astronauts. So we undertook a study called MicroAge, and this was funded by the UK Space Agency and undertaken in collaboration with Kaiser Space, our integration partners, and the European Space Agency. So we know that inactivity is bad for muscles. What can we learn from the extreme conditions? Basically, the astronauts, even though they exercise, their muscles think that they are extreme couch potatoes. They're not really moving. First problem that we encountered was that there are very few astronauts in terms of power for studies on the space station. We wanted to do a lot more studies, so we wanted to send up 3D muscle constructs so we could improve our N number. So we did this and got our N number in this situation up to 72. We took human muscle immortal cells, we fused them to form myotubes, and then they form 3D muscle constructs. You can see very roughly here, you can see there are electrodes across the muscle, so we can put a voltage across and we can make the muscle contract. Now I'm hoping that the video may not run in this presentation mode. Maybe it does. Oh yes, it does. So you can see here we have quite a slow protocol. If you focus on the top of the construct, you can see there is some contraction going on there. So we can make the muscles exercise using any protocol we want. So the next challenge we had, and again, I don't have much time to go through this in any great detail today. It's all been published and the publication links are in the slides. But on the left, you can see this is mostly Aurora scientific kit. And one of our engineers, Kai Herker, this is the extent of the kit that the equipment that we would normally use to undertake the studies we wanted to do. We needed to miniaturize that into something the size of a pack of cards or a mobile phone. And our engineers managed to do that. The system also needed to be plug and play. It needed to be plugged in and run autonomously. We needed to reduce the size and the weight. We needed to keep the muscles alive with oxygen, media, temperature. We needed to run an experimental timeline, which was controlled under the PCBs, fluid exchange, stimulation to contract. Because of the anabolic resistance, we needed to also know that they had contracted and that there wasn't just a failure in the system, that we didn't see a response to contractions. And we did that by measuring impedance, and we recorded the results. There are lots of safety issues and environmental issues as well that we had to overcome. There was lots of novel engineering with microcontrollers, miniature pumps, 3D printing components, gas permeable membranes, and extensive temperature and vibration testing. We also wanted our muscle constructs as mature as possible. And so we undertook a study to look at the best time point to look at these constructs. And these data are published quite recently by Ben Tollett in our lab. And we focused on day 8, which is a point where we have optimal production of muscle proteins, and we can also measure muscle contraction. And so this just shows you that we can measure force generation in the constructs. This is an activation of contraction. The protocol that we used was non-damaging because we could repeat it time after time and didn't get much in the way of reduction of force for the protocol that we used. And also there's a linear response. We have improved these data since, but a linear response with force versus impedance measurements. So the study that we undertook in microgravity was that we grew these constructs, we integrated them into the system that I've just shown you, and they were then launched on SpaceX-24, integrated in an incubator on the ISS into the incubator called Cubic. In the protocol, we had, with and without a centrifuge, you'll see this in a minute, within the Cubic, which gives a 1G control in there. We electrically stimulated some of the muscle constructs to contract. Interestingly, we also had HSP10 transduced muscle constructs in there as well. And the reason for that was because, as I showed you earlier, we'd seen some evidence of preservation of muscle function with genetically modified mice overexpressing HSP10. So that was one of the interventions that we tried. These muscle constructs are about the size of a grain of rice, but we managed to undertake, because of the power of the study, transcriptomics, proteomics, secretomics, looking at cytokine release, and of course impedance. And we did a comparison to what happens with aging. This is where Vector Builder came in and produced a really reliable lentiviral plasmid here that you can see with an EGFP reporter gene, blastocidin resistance gene. And we did some studies to look at optimal HSP10 overexpression. We're very happy with the levels of expression that we got. And we landed on an MOI of 100 for 144 hours with greater than 80% efficiency, more usually up to 90 odd percent. And again, these data are all published as a preprint at the moment. So we sent our team to the International Space Station processing facility. It was during COVID, or just at the end of COVID, hence the masks. So produced a number of other challenges, as if we didn't have enough. We built the constructs. They were there for 6 weeks, building 100 constructs per batch, and we had to have reserves in case there was a scrub launch, 2 sets of reserves. So we had about 400 constructs in the incubator at any one time. We integrated them into the containers, signed them over, the kit containers, and then NASA came and picked them up. This is the team. The rocket, SpaceX-21, is just behind here. And our samples are in the top Dragon capsule there. You can just about see it in more detail here. So that happened 2 days before the launch. And then we launched. They were taken to the space station. I think this video should be running. And then unpacked. You can see that in a temperature-controlled box here. And Matthias Maurer is unpacking the constructs. and putting them into their rightful positions in the incubator. And you can see the centrifuge in the middle here for some of the controls. You can also see one of them floating off here, which he lost for a little while, but they did manage to find it and put it back into the incubator. So our main questions coming back to muscle loss is, does extreme inactivity result in muscle changes similar to those that we see in older people on Earth? And if so, does it include an attenuated adaptation to contraction? So the two major issues that we see on Earth. I'll cut to the chase immediately and say, yes, it does, but in a much more rapid timeline. So muscle constructs, these data show if relative to ground, what happens on the ground or on Earth, the levels would be 100% if it was the same as Earth. So what we see in terms of cytokine release by these muscles is, and chemokine and growth factors, so you can see a reduction in some of the PDGF, VEGF, but a significant increase in TNF-alpha release, RANTES, MCP1, after just 48 hours. Not good news for the astronauts. Very similar to what we see in quiescent old muscle with this activation of NF-kappa B. So what happens following acute stress? In this instance, we want acute increases in some of the other cytokines, but they will fall off very quickly. So we need that for adaptation. This is the ground control. And so we're looking at the effect of contraction versus quiescent muscles. And we see increases, quite variable, but increases in some of the, in this case, some of the growth factors and so on ground. Very briefly, when we undertake that in microgravity, most of these are reduced. You can see IL-2 has gone completely, as has VEGF in microgravity following contractions. So we've got some major changes there in the adaptive responses in terms of secreted cytokines. I don't have time again to talk you through the whole of the proteomic sets of data. And again, we have it's the same preprint up at the moment, and we're just about to submit this for publication. But in summary, the effect of microgravity on the proteome of 3D muscle constructs is that we saw a downregulation of the key proteins involved in energy metabolism and stress responses, the HSPs, structural integrity, and an upregulation of catabolic and apoptotic enzymes. So really not good for the muscle, and in a very similar manner to what we see in old age. When we look at the overall protection of HSP10, that's quite dramatic. And what we saw was the overexpression of HSP10 attenuated the effects of microgravity, particularly preserving mitochondrial protein expression, particularly in the matrix, promoting mitochondrial gene expression in microgravity. So really preserving mitochondria. And that leads on to MicroAge II, which we're nearly ready for launch with. MicroAge II will focus on mitochondria and look at mitochondria as gravity sensors in microgravity or activity sensors on Earth. This system allows us to test interventions that may take decades to test on Earth. Muscle loss occurs at a rate of about 3 or 4% on Earth in older people. So there's a lot of noise in those data. So you need a long time to study them. Here we've activated the processes very rapidly so we can test a number of interventions. And that's our plan in MicroAge II and some of our other funded studies that we have in microgravity too. Just like to take the last minute or two to tell you about a little bit of our outreach. So outreach is incredibly important. We've spent years and years working on aging and exercise, and we've tried to get children engaged in this, and it's really difficult. And nutrition as well. They don't think they're ever going to age, and they think they exercise enough. But if you then start to introduce space and microgravity, they are desperate to engage. And so we did a number of things. We had an app. We had a patch competition where the winning patch was adapted by a graphic designer and made into our patch. You can see me wearing it here. We had live Zoom calls with the children. 24 of them were shortlisted and they were sent up to space inside our kit containers. and they got them back at the end. We also work particularly in Liverpool, but with other regions as well, and have advised across to the US as well in terms of working with children in disadvantaged, particularly situations. So you can see here the children are exercising like an astronaut. They wouldn't exercise without the astronaut, but they're exercising like an astronaut and undertaking a whole host of things. You can see there, you may be able to hear the sound, but they're building rockets here as well. And there goes the rocket. So in summary, data suggests that we have a model of accelerating muscle aging, at least lots of the components. Aging is associated with attenuated responses to contractions during, and that has a functional importance. Microgravity has really allowed us to understand muscle loss during aging, with mitochondria being core to this muscle loss, acting as activity sensors. And preservation of production of HSP10 in the mitochondria provides substantial protection both during aging and in microgravity. It just remains for me to acknowledge the multidisciplinary MicroAge team. So these are the core people involved at the University of Liverpool, particularly Malcolm Jackson, who's a co-PI, Sam Jones, who we couldn't run without. She's our program manager. Kai Herkes, who's our lead microelectronics engineer, and Chris also, David Turner, who works on our imaging and microscopy, and then the whole team that undertake the laboratory analysis, particularly our proteomics facility, Kaiser Space, our integration partners, and ESA and UK Space Agency, and our funders, of which there are many. Thank you.
Thank you so much. That was very, very cool.
Thank you.
All right, we have some questions. We can get started with the first one. I assume NASA has a post-return training program for astronauts. Do you plan to examine muscle redevelopment in that context?
Great question. We would love to, and some of the data are published in this instance. It's quite tricky to get access to those data. It's really difficult to know what happens to those muscles when astronauts come back, because if they've lost whole muscle fibers, they will never get those back. But they can build what fibers they've got left and make them bigger. And so it's almost impossible to know which they've done. Have they lost muscle fibers, or have they just made what they had before they went up bigger? And so they can get that muscle mass back. What we'd really like to do is to look at older astronauts to see how their muscles age. But yeah, we are keen to look at all of the data that's available from astronauts, particularly related to muscle.
Thank you so much. We have another question. Have you had a chance to look at how great the effects of exercise, like weightlifting, etc., has on slowing muscle loss? How much do people need to do to have a significant impact?
The level is really difficult. So I think the latest, and it's some of our active research physiotherapists suggest that you really need quite a relatively high level of exercise to have an effect. And so some gardening, weekend gardening or whatever isn't enough, that you really need. And one of the quotes I was told recently was, you need exercise that increases your heart rate and really, makes you work a little bit. Obviously, with the recommendation that any sort of exercise you need to talk to your clinicians and GP first. But it doesn't slow that rate of loss of muscle strength, no matter how much exercise you do. All you do is optimize what you've got left. We think that rate of loss that you see, that what affects that rate of loss is loss of the fibers, and we're trying to understand, and we're starting to understand why we lose muscle fibers. There you see you do start to see denervation of individual fibers as we get older. So there are active studies looking at that, but we can't intervene in that fiber loss at the moment.
Thank you so much. One of our attendees is wondering if there is a gender difference.
The numbers are too low. Yes, we would be very keen to look at that. Numbers are too low with astronauts at the moment. We don't have enough female astronauts, but it would be good to see if anything sticks out there in terms of that. What we can do is, so the muscle fibers we sent up were from an adult male, 21-year-old, I think, 21-year-old European. And so we're very keen to look at the effect of whether there are any gender differences, even just using the source of our immortal cells from different genders and different nationalities as well. So we're keen to do that.
Cool, thank you. So related to the first question that was asked regarding the NASA and the post-return training program, we have a follow-up question. For the aging-like changes, that you observed under microgravity, are they reversible once normal conditions are restored? Do they have any plan on, do you
In
know?
our constructs, we would love to know that, what the it would be amazing to look at the effect of that when they got back onto Earth, and that would be quite a tricky experiment, because what we do is send our muscle constructs up to microgravity. The ones for MicroAge II were actually up there for 42 days. At the end of the two or 3 day experiment, they were frozen on the space station. So we would have to get them down live and then see their effects. That's not to say that that's not going to become possible soon. There are options of sending up muscle constructs potentially on satellites and having return, and for other systems. So more rapid turnaround of samples going up and coming back down would really allow us to do that. But really interesting question. Yeah.
Cool. Can you quickly expand on what type of cells, donor, age, etc. these were? Since these are immortalized, the bioenergetics are probably quite different from human muscle or human primary cells. Are you concerned about this regarding the mitochondrial focus in the next mission?
We're not particularly concerned. So we know that these muscle constructs behave like they replicate the responses to contractions on Earth that we would normally see, and they replicate a lot of the changes that we would see in muscles in vivo as well. So we're not concerned about that. But obviously, it's a model system, so it will have its limitations. Primary cells produce their own challenges, the variability being the biggest of them. And so we are looking as well at potential use of primary cells, but too variable at the moment, particularly for the limitation of the numbers that we have.
Thank you. We have two related questions. The first one being, for the muscle fibers grown in the incubator in the space station, are they grown in physiologically equivalent conditions? And somebody is also wondering, why not just use microgravity environment on Earth? Easier.
Okay, so we try and mimic. We have as many controls in the system as possible on the space station in the experiment. So they're maintained at 37 degrees. We know that they get sufficient oxygen with the systems that we use, sufficient media changes, and so on. But we also have controls in there. So our 1G samples that were in the centrifuge within the incubator would control for everything but the change in gravity. The minor issue we have with that is that the cells are in microgravity within a rapid time after launch, and they're not put into the centrifuge until a little bit later. So there is a limitation to interpretation of the use of centrifuge, but at least that controls for other concerns that might be, such as radiation and so on. And sorry, what was the second part of the question?
Have you tried, why not use microgravity environment on Earth?
Yes, so random positioning machines and so on. We are doing some of those studies, and again, they really have their limitations. Certainly, potential of shear stress and so on. So, vector changes, it may not mimic what we see with complete unloading, but we are looking at those potential systems. But we've been a little disappointed with some of the preliminary studies that we've done so far.
We have another question. In your proteomics data, we see an interesting lower abundance of proteins needed to break down glycogen. Is it known whether the changes in muscle function or mass with age could be related to an inability to use glycogen for energy? Have you tried to rescue this downregulation in your system?
Interesting observation. No, we haven't tried to. We haven't looked at that. So it is an interesting observation that we'll certainly take a look at.
Awesome. Apart from complete muscle loss, is there a point of no return with the rate of loss?
No. um, that's very hard to say. Certainly in old age, we see, 80, 90-year-old people have lost 50% of their muscle mass. But, if you've undertaken lifelong exercise and you've maintained the best function for the mass you've got left, then, their point of threshold where they need care, need help, it is maybe 10, 20 years later. In terms of microgravity, there's an acute loss. I mean, obviously, if they lose 30% of their muscle on average, 20 to 30% by the time they return. A lot of that is lost within the first few weeks, and so it does taper off a little bit. So what we're looking at is that acute loss.
Okay. Somebody is wondering if you've considered using an in vivo model to investigate whether the aging-like changes induced by microgravity are also observed at the organismal level.
Lots of people have sent rodents up to the space station and other systems such as nematode, and they do see similar losses in muscle mass and function. So.
Okay. I believe we are reaching the end of our questions, maybe one or two last ones. How do experiments play out in the context of existing NF-kappa B mouse models, both knockout and overexpression models?
Yeah, we are a little confused about the different models now, and I think the difference is that what I tried to stress in my talk, which was we need that acute activation of NF-kappa B to produce some of the positive, the beneficial proteins that are used during hypertrophy. And so we need that, but we don't want that to be chronically activated. And what we're doing now is really working with, I think I mentioned David Turner earlier, but what we're looking at is the subtle difference, not only in NF-kappa B activation, protein compositions, but the timing of cycling into the nucleus and back out and so on. Lots of which control transcription of different genes, and we're trying to tease that out. That's a substantial project on its own, but a really interesting focus.
Okay, wonderful. You kind of answered this already, but we'll make this our last question just to like up. Is there any strategy to reactivate muscle satellite cells to help counteract age-related muscle loss?
Yeah, satellite cells are a really interesting challenge. So if you take out satellite cells from muscles of old mammals, then they will behave perfectly well. We have enough satellite cells in a healthy individual to rebuild muscle. The problem is that it's the environment that they are in, and so particularly this chronic inflammatory environment and the niche that the satellite cell sits in is a potential problem. And we and others at University of Michigan have undertaken studies where if you take muscle from an adult mouse and transplant it into an old mouse and vice versa, the muscle it degenerates and regenerates spontaneously, and it behaves like the age of the host. So a young muscle in an old mouse will behave like an old muscle. So we really need to try and deal with the environment that the satellite cell is in.
Okay, we do have one more question that just came in. In terms of the plateauing of muscle loss in microgravity, do you hypothesize that there is a maximal amount of muscle loss beyond which further degradation will not occur?
That's a possibility. Yes, we don't know. We haven't studied muscles of that in that. So it would be really interesting to look at muscles that have been constructs that have been up there for that length of time without intervention, so we can really map out what happens during those later time points.
Amazing. Thank you so much. There's also tons of messages here talking about how everybody enjoyed the talk and how wonderful.
Oh, thank you.
Oh, that sentiment as well. It was really great, and we are very excited to see what your next mission brings, and hope you it and hear about it soon.
Thank you. Thank you for the invitation to talk. And if anybody wants to ask any more questions, please do just reach out.
Excellent. Thanks, everybody. Thank you for attending this talk, and we hope to see you on our next one.